Hello everyone, this is LATHA AKULA. In this video, we are going to study about the last concept in the enzymes, that is nomenclature, classification and chemical nature of the enzymes. In this first one is the nomenclature. This nomenclature was proposed by International Union of Biochemistry IUB. They have appointed an enzyme commission in the year 1961. That enzyme commission, they devised some basic principles for the classification and nomenclature of enzymes. According to those principles, that IUB, they have divided enzymes into 6 major classes, and each class is again subdivided into many subclasses, and which are further divided into subclasses and etc. And here for enzymes, this IUB, they have given 4-digit enzyme commission number is assigned. For example, if we take an example 5217, this is one enzyme commission number. In that, the first digit 5, that denotes the class of enzyme, and second digit 2, that denotes the subclass of enzyme, and the third digit 1 denotes the subclass of enzyme, and finally 7 denotes the individual enzymes. This is about the nomenclature of enzymes, and now we'll see the classification of enzymes. The enzymes are classified into 6 classes. In that, the first one is oxidoreductase or dehydrogenase enzyme. So this oxidoreductase enzyme, they catalyze oxidation reduction reactions. Reactions. In that, what will happen? The transfer of electrons or protons from one molecule to another molecule. In that, the molecule which lost the electrons are called as oxidized condition, and the compound which received the electrons, that is called as in reduced condition. Examples for these oxidoreductase enzymes were dehydrogenase, oxidase, reductase, catalase and peroxidase enzymes are the examples for oxidoreductase enzymes. Second one is transferase enzyme. The name itself indicates what is the function of this enzyme. They transfer several specific groups from one substrate to another substrate. So what is the function of these transferase enzymes? They catalyze the transfer of specific groups from one substrate to the another substrate. Examples for these transferase enzymes are transaminase and kinase. Here, the transaminase that transferred the amino group from one substrate to another substrate, and kinase that catalyzed the phosphorylation of substrate. What is its function? That they transfer phosphate group. Here the phosphorylation of the substrate by transferring the phosphate group. From where it is transferring the phosphate? By using the ATP. ATP contains 3-phosphate, right? So this kinase enzyme, they transfer the phosphate groups from the ATP to the substrate. If we take the example, this glucose is present here. Glucose is combined with ATP in the presence of glucohexokinase. Here what will happen? This enzyme transferred the phosphate group from the ATP to the glucose. Here ATP contains, adenosine triphosphate contains 3 phosphates. Here this enzyme transfers 1 phosphate from this ATP to the glucose. Then glucose will become glucose 6-phosphate, where it is added in the 6th carbon place, the phosphate group is added and it will become the glucose 6-phosphate, and finally ATP, adenosine triphosphate, by losing 1 phosphate will be converted into the ADP. This is the function of transferase enzymes. The next one is hydrolases. So what is the function of these hydrolases? They break down large molecule into the smaller molecule by the addition of water. By using water, they break down the complex substrates into the simpler ones. Example, most of the digestive enzymes, almost all the digestive enzymes will come under this, like protease, amylase, lipase, maltase, nuclease. So all are the digestive enzymes, right? They will come under these hydrolases. If we see sucrose is a disaccharide. It contains 2 monosaccharides like glucose and fructose. So here, this disaccharide glucose is broken down into monosaccharides by using water in the presence of an enzyme, sucrase or invertase, and finally it will be broken down into the simpler, smaller substrates like glucose and fructose. Next, fourth class of enzymes are lyases. So these lyases, they also, like hydrolases, they also cleavage the substrate into two parts. But here there is no use of water. In hydrolases we have studied the reaction occurs in the presence of water. But here in the absence of water, without the use of water, the complex substrate molecules was broken down or cleaved into two parts. And examples for this lyases are aldolase, decarboxylase and carbonic anhydrase. Aldolase, decarboxylase and carbonic anhydrases are the examples of lyases enzymes. And here there is a formation of double bond also occurs here. If you see the equation, fructose 1,6-diphosphate in the presence of aldolase enzyme, this is the complex substrate which is broken, which is cleaved into two substrate, that is dihydroxyacetone phosphate and glyceraldehyde phosphate. So fructose 1,6-phosphate is broken down into dihydroxyacetone phosphate and glyceraldehyde diphosphate in the presence of a lyase enzyme, that is aldolase. The next one is isomerases. So isomerases are nothing but these are the enzymes. What is their function? They catalyze the rearrangement of molecular structure. So here these isomers are the they have the similar molecular formula, but these enzymes, they rearrange their atoms and groups. Here these enzymes, they catalyze the rearrangement of molecular structures to form isomers are formed. So here these isomers are they have the same type of molecular formula is present, but their arrangement of atoms and groups will vary. Example for this is the isomerase enzyme. This isomerase enzyme that will convert the glucose 6-phosphate into the fructose 6-phosphate. The next one is ligases. So up to now we have studied that some enzymes, they will break the substrate into the two. But here these ligases, what is their function? Here these enzymes, they catalyze, they will combine two substrate molecules. They form a covalent bond between the two substrate molecules by using enzyme. Two substrate molecules are combined very strongly by using ATP molecule. Examples for these ligase enzymes are RUBP carboxylase, phosphoenol pyruvate and ligase are the main examples for these ligase enzymes. And if you see the equation, phosphoenol pyruvate is combined with carbon dioxide. So here the carbon dioxide is attached to the phosphoenol pyruvate by using ATP in the presence of an enzyme phosphoenol pyruvate carboxylase, and finally it will be converted into oxaloacetic acid. When the phosphoenol pyruvate is combined with carbon dioxide, it will convert it into oxaloacetic acid. So these are the 6 classes of enzymes. So now you will see the chemical nature of enzymes. We know that most of enzymes are the tertiary proteins only. These enzymes are two types. They were simple enzymes and conjugated enzymes. If you see the simple enzymes, they consist of, here the protein alone will act as enzymes, that is called as simple enzymes. Here the protein will acting as an enzyme. There is no non-protein part is present in this. Example, trypsin and pepsin are the example for the simple enzymes. And what are the conjugated enzymes? Here there is a protein part is present. In addition to the protein part, there is another non-protein part is also present. Protein part and non-protein part, if two are present, that is called as conjugated enzyme. In simple enzymes, only protein part is present. The protein alone will act as an enzyme. If we see the protein part of this conjugated enzyme is called as apoenzyme, and the non-protein part of the conjugated enzyme is called as cofactor. Here, this apoenzyme is non-functional in nature. Alone, without cofactor, the apoenzyme will not catalyze any reaction. But when this non-functional apoenzyme, which is a protein, it is combined with the cofactor, then it will become the functional enzyme. Then it will become the functional enzyme, and this functional enzyme is called as holoenzyme. What is the holoenzyme? When the protein part and the non-protein part will combine, that will form the functional enzyme, that is called as holoenzyme. Now you'll see the types of cofactors. So we have studied that the non-protein part of the enzyme, which is the important one. Here, these cofactors are of two types. Some are organic, some are inorganic. The organic non-protein part and the inorganic metallic ions. If you see the organic non-protein part, depending upon the separation, there are two types. Some non-protein parts, they can be easily separated. That means they were attached to the apoenzyme temporarily attached. Those enzymes are called as coenzyme. They can be easily separated. And for these coenzymes, for their action, vitamins are necessary. Examples for those are NAD, NADP and FAD. FAD means flavin adenine dinucleotide. And no, and some non-protein parts are, they cannot be easily separated. That means they have permanently attached to the enzyme, apoenzyme. Those permanently attached, they form the covalent bond with the apoenzyme. They are called as prosthetic group. Example for the prosthetic group is heme. And the last one is inorganic metal ions. These inorganic metal ions, they will act as the activators. They will act as the activators for the enzymes. Examples for these activators are the several metal ions like zinc, copper, iron, calcium, magnesium, copper, cobalt, molybdenum, manganese, so chlorine. All these are the several activators. In that, the zinc will act as a it is an activator for proteolytic enzyme like carboxypeptidase, and copper will act as activator for the cytochrome oxidase enzyme. Here, if the cofactor is removed from the enzyme, the catalytic activity of the enzyme will be lost. That means the cofactor plays a crucial role in the activity of the enzyme. So with this, we have completed the topic enzymes in the biomolecules. And if you like my video, please like, share and subscribe to my channel LATHA AKULA Biology Tutorial. Please keep watching.